Dear students, dear colleagues, I again thanks for the National Academy of Science Education for the prize, and I would like to share the cornerstones of this discovery with you. So this year, there was a famous event in New York. A long quest of consciousness seems to be ended because 25 years ago, two eminent scientists made a bet. Christoph Koch, the neuroscientist, said that from 25 years later, which was this year, the neurological basis of consciousness will be revealed, whereas David Chalmers, the philosopher, said that it will not. And among a large media attention, the two came together in New York, and you may guess what was the result of the bet. Obviously, the neurological background of consciousness still is not known. So Christoph Koch lost, and this cost him at least 25 bottles of very expensive red wine. Nevertheless, we have many, many different ideas and theories of consciousness. And as you can see, this is the human brain from the side with its convoluted surface, which is called cortex. And there are various dots and arrows. So one thing is common in these theories, that all of them links the emergence of consciousness to various sort of cortical activity. So no subcortical input is shown here, and as Christopher Koch basically stated, any subcortical input is basically just permissive. So it's not actively involved in generating consciousness, rather it just generates the state where it can occur. If we go back in time, a couple of years, there was a big review in Science, the controversial correlates of consciousness. You may observe that it shows similar dots and arrows, but there is one difference here. It shows an input to the system, and this is the thalamic input. You may appreciate that it reaches only the very beginning, the first step of the entire process. Still, cortical-cortical communication is responsible for the emergence of consciousness. Okay, so we may basically transform this view into this very simple scheme. You have a brainstem which generates all the information from the brain. It wires through the thalamus, it sends it to the cortex, and then cortical-cortical combination achieves consciousness, which is then projected back as a behavioral output. The only problem with this view that it is not true. So many, many researchers have been studying this question, and it's clear now that each and every cortical region is connected to the thalamus, not only the sensory regions. So motor regions, regions connected to memory formation or the representation of our inner states and emotions, they are all connected. And this connection is bidirectional. So not only thalamus projects to the cortex, but cortex projects back to the thalamus. What's more, thalamus appears to be a unique bottleneck of information, since with the exception of olfactory bulb, almost all fast glutamatergic inputs reach the cortex via this thalamic transfer. So cortex is, in a sense, isolated from the rest of the world. There are slow communication between the brainstem and the cortex, but everything that is important has to grow through the thalamus. So where is the thalamus, and how we can study it? Thalamus is in the heart of the brain. It's in the very middle, and if you make a cross-section in the thalamus, this is what you get. This entire large stuff is the cortex, the gray matter, which contains the neurons, and the white matters, which contains the billions of axons linking cortical regions. And in the middle, you have the thalamus. now, this is how thalamus looks like in a tissue preparation. This would be dorsal. This is half of the thalamus. This is medial. And if we zoom into this picture, then I say zoom into this picture. Okay. It doesn't want to zoom in. Okay. Then you will see that thalamus indeed receive huge amount of axon terminals, and these are the axon terminals which carry information from the outside world and to be relayed to the cortex. But the critical thing is that thalamus and cortex is bidirectionally connected. So lesson number 1, thalamus and cortex form one functional unit, and if you want to understand normal or pathological activity, you have to understand how they communicate. And indeed, many, many diseases linked to the alteration in cortical activity. So if you want to cure them, you have to know its neurological basis. So János Szentagottai, the godfather of Hungarian neuroscience, made a very significant contribution how we understand neuronal organization. He launched what we call a modular concept in neuroscience. It basically states that you can understand the brain as composed of individual units. In case of the cortex, he called it a cortical column. A cortical column has a quite complex internal organization consisting of many, many dozens of cell types, but it's approximately 800 to 1,000 micron wide. But these cortical columns are basically identical. So we have a modular organization. You have lots of modules which are very similar and linked to each other. It is very easy to understand the advantages of this organization. It's much more easy to code for the development and or for the evolution. If you want a bigger brain, you simply have to add more modules. And obviously, you always have a question in their mind. What if not all units are identical? What if there are differences between the way how cortex communicates with the thalamus? So we set out to investigate this question, but before I tell you the result, I have to introduce you another structure. And this is a thin sheet of neuron which entirely consists of inhibitory neurons. Inhibition is extremely important in the brain. It not only dampens activity, but it organizes the activity. It filters out unnecessary information, and also it helps to synchronize neuronal activity. So TRN provides the major input to thalamus, and without TRN, there is chaos there. So we set out to examine whether the connectivity between cortex and TRN is uniform throughout the cortex. We used transgenic cell mice in which you can label selectively individual cell types, in this case, the layer 5 of the cortex. So first we injected into the somatosensory cortex, and this is what we found. The axons went through the TRN or reticular nucleus without giving of collateral, so basically there were no communication between cortex and thalamus. Apparently, this is a direct excitation without an inhibitory interface. So we repeated the same experiment by injecting into frontal cortical areas, which are responsible for movement, organization, planning, and other higher order cognitive processes. And this is what we found. Huge amount of collaterals innervating the reticular thalamic nuclei cells. So just as a cartoon, you can see here lots of collaterals. So all the information which goes through the thalamus is also related to the TRN. I have to pause here because when we first saw these sections, we were basically stunned. It was completely unexpected. We know about this lack of collaterals, but what we saw, this was a real discovery when you immediately understood that you discovered something completely novel which was to study. And indeed, we made many, many experiments. We examined the ultrastructure of these terminals. Here you can see a synapse between the cortical fibers and the TRN neuron. And we also studied the physiology of this pathway. We can selectively activate this pathway using a method called optogenetics. And the interesting thing we found that with increasing intensity, with increasing stimulation intensity, these are the neuronal response of excitation action potentials. So as we increase the intensity, the TRN neurons responded with higher frequency action potential and more action potential. So basically, there was a significant correlation between the laser power and the response frequency. So we can conclude that this highly specific cortical impulse generates an inhibitory signal in the thalamus, this TRN projects to the thalamus, that precisely match cortical activity, but only in the frontal cortex and not in sensory cortices. Okay, so what have we learned so far? This is a beautiful TRN cell surrounded by the layer 5 terminals. So we learned that in case of sensory cortex, there is no collateral between layer 5 and TRN, and it's quite distinct in case of frontal cortices. Obviously, we have one question remaining. What about the direct connection between cortex and thalamus? Can it be also different? And this is what we set out. We are just finishing the experiments right now. This is exactly the same experiment, but now we study the thalamus, not the TRN. So when we inject the somatosensory cortex, we see giant excitatory terminals in the thalamus, which I can represent here. These terminals are pretty powerful and can really drive thalamic activity based on cortical activity. When we examine the thalamus after frontal cortical injection, the difference is also very evident. Here, in this case, you only have small terminals. So apparently, not only the collaterals in the TRN, but the direct connection between cortex and thalamus also distinguishes these two corticothalamic pathways. Obviously, if this is small, then we have to identify whether it's still successful. So again, we activated the frontal cortical activity and the recorded neuronal activity in the thalamus. These are individual laser flashes which activate the cortex, and these are action potentials generated by thalamic neurons. And as you can see, with a very brief delay, less than 10 milliseconds, you have a reliable action potential generation in the thalamus. So we can conclude that even though these terminals are small, they can faithfully activate thalamic neurons. At the electron microscopic level, the two terminal types are also very different. The big one formed many, many synapses with the thalamus with this complex postsynaptic surface. But in case of the soul, we notice something quite unusual. If you look carefully, this postsynaptic structure, this is the presynaptic terminals with the vesicles, which is released and activate the postsynaptic cells. It has a peculiar shape. It has a neck and a head. And this structure is well known in neuroscience. It is called a spine. But spines has not been described in the thalamus so far, so we're pretty much surprised. You can see the same thing in light microscopy. And the strange thing about spine is that dendritic spines in the brain are the critical structures of synaptic plasticity. And plastic changes in synaptic strands are the cornerstones of forming new memories. So what we are approaching here is that it's not only that the two pathways are pretty different, but frontal corticothalamic pathways can be plastic. So let me summarize my talk. The first conclusion is that thalamus cortex forms one functional unit, and thalamus is apparently involved in every cortical cooperation. So if you want to do any specific postulates for, say, conscious behavior, you always have to involve at least one or two arrows for the thalamus. Second, apparently the cortical control of the thalamus is region-specific. The wiring diagram is qualitatively different between these two regions, which means that distinct operation will take place here. Frontal thalamocortical pathways can be plastic, and obviously, if you want to understand the neuronal underpinnings of this disease, especially those ones which involve the frontal cortex, you have to acknowledge the specificity of these neuronal circuits. And with this, I would like to say thank you for your attention and for my entire research group, in particular Hardinger Nora, who is the first author in the awarded paper, Boglárka Tóth, who is sitting among you and contributed significantly to both studies, as well as to my wife, Hajnaka Bokor, who made physiological recordings in the second experiment. And thank you for your attention.